Okay, thank you for the introduction. Welcome to the SBIA meeting. My name is Keaton. I'm the Associate Director in Division of Bioequivalence 3, Office of Bioequivalence in OGD. For the next 30 minutes, I will share several case studies for inadequate BE study from the regulatory perspectives. Here is my outline I plan to cover. First, a brief overview of the regulatory criteria for in vivo BE studies. Next, a summary of common BE deficiencies in pharmacokinetic studies. Next, several case studies in details. I will provide case studies in 3 categories. First, study design, Tlag difference, and the point estimate for reference scaling BE approach. For the study design, I will cover some cases on the insufficient sampling time to capture Cmax or unable to calculate partial AUC. Also, I will cover some case study on the insufficient washout period during the study. At the end, I will wrap up with a brief summary. So first, we'll give an overview of the regulatory criteria for in vivo BE studies. For general criteria for adequate in vivo BE studies, the number of subjects in the study should be sufficient for BE determination. A minimum of 12 subjects should be included in any study to provide a appropriate representative for general populations. For both single dose and steady state studies, a 19% of confidence interval for Cmax and AUC should meet 80 to 125% criteria. Although there's no formal statistical test for Tmax is required, the median Tmax and the Tlag between the test and the reference products should be comparable. In addition to the general BE criteria, there are specific criteria for lethal therapeutic drugs and highly valuable drugs. It is not our scope to discuss what is the classification criteria for NTI and highly valuable drugs for this talk. For NTI drugs, there are 3 BE limits. 1, the BE limit for PK parameter based on reference scale BE limit. 2, the BE limit of PK parameter based on average BE with 80 to 125%. The third one, the upper limit of 90% of the ratio of the within subject standard deviation of the test and the reference is less than or equal to 2.5. So the agency also chose warfarin tablets as a model drug to include stepwise procedure in this product-specific guidance. For highly valuable drugs, there are 2 BE limits. One is the reference scale limit. The second one is the point estimate constraint of the TR geometrical mean ratio should within 0.8 to 1.25. The stepwise procedure is also included in the product-specific guidance for progesterone capsules. So here's a link for the calculation. For an adequate BE study, the sampling schedule should include frequently the sampling around the predicted T maxima to provide a reliable estimate the peak of the exposure. Particularly, the sampling schedule should be planned to avoid the same maxima in the first concentration time curve. Also, the sampling schedule should cover concentration time curve longer enough to provide a reliable estimate of extent of drug exposure, which is achieved if AUC 0 to T covers 80% of AUC infinity. At least 3 to 4 sample times should be required during the terminal log phase in order to reliable estimate of the elimination rate constant. For the study design recommendation, please refer to BE studies with PK endpoints for drugs submitted under NDA guidance. Please also check recommendation in the product-specific guidance. If the applicants proposed new approaches which are deviate from the product-specific guidance, please include sufficient justifications in your submission. The acceptable on the BE study is evaluated on a case-by-case basis. The applicant also can submit control correspondence to clarify the study design. For complex drug product, the applicant may submit pre-NDA submission to propose new study and study design. FDA recommends a single dose, two-way, two-treatment, two-period, two-sequence crossover study for most drug dosage. That drug release intend to systemic available. However, crossover study may not practical for the drugs with longer half-life, like more than 24 hours. In such case, the applicants can choose heteroline design, where the two treatments were dosed to our separate groups of subjects with similar demographics. Next, we will discuss common BE deficiencies in PK studies. The BE deficiencies can be divided into 6 categories. 1, analytical issues. It includes pre-study method validation and within the study. 2, inadequate study design. 3, inappropriate statistical approach. 4, incompletely SAS transport files. 5, failed to reserve study samples. 6, data integrity of the clinical and analytical sites. In the next slide, I will share 7 cases in our review process. Case 1, these studies was conducted in 2-way crossover instead of 4-way fully replicate study for the following categories. 1, NTI drugs. Since, as mentioned earlier, since we need to compare within subject variability between test and the reference product, 4-way crossover should be used in BE studies. 2, like dabigatran capsules, it has demonstrated a steep exposure response relationship for both safety and efficacy. Therefore, the reference scale BE should not be used to widen the BE limit. The applicant should use average BE with 80 to 125% criteria. Also, we did compare within subject variability between test and reference products. So the upper bound of 90% of the TI ratio of the within subject variability should be less or within 2.5. 3, for some modified release drug products like methylphenidate ER tablets, to ensure the switchability between the catheter and the generic drugs. Subject by formulation interaction for each PK parameter are recommended in addition to the establishment of average BE. So here is a case 2. Insufficient sampling time to capture C maxima. The T maxima of drug A is about 0.5 hours. 41 out of 46 subjects had the first post-dose sampling time 0.5 hours as C maxima. It raised a question of bias of estimating of C maxima because of insufficient sampling time at the early sampling schedule. Per PK endpoint guidance, failure to include early sampling time, like between 5 to 50 minutes, leads to the first sampling time C maxima, may lead FDA not to consider the data from the affected subject from the data analysis. So case 3, insufficient sampling time at early partial AUC. Per product specific drug guidance on drug C, we recommend partial AUC in BE evaluation. For this one, we will recommend partial AUC 0 to 3 and 3 to T. At least 4 long zero measurement of concentration are recommended for each partial AUC. However, the applicant only include 2 sampling time for the partial AUC 0 to 3, which is 0 hour and 2 hours. So this sampling time for this study are not sufficient to accurately estimate partial AUC. The applicant was asked to repeat the study with adequate number of the blood sampling in order to reliably estimate partial AUC. So here is the standard 2-week crossover BE study scheme. There are 2 treatments, test and reference. The treatment period should be separated by a washout period sufficient to ensure that the drug concentration are below the LOQ for all the subjects at the beginning of the period 2. Normally, at least 5 half-life are necessary to achieve this. So here's the case study 4. The half-life for drug B is greater than 100 hours. T maxima is about 1.5 hours. The applicant chose the 15 days as a washout period. 43 out of 44 subjects had pre-dose concentration in period 2. Among them, 35 subjects had a pre-dose concentration greater than 5% of their respective Cmax at period 2. Based on PK endpoint guidance, we recommended that if pre-dose concentration is greater than 5% of the Cmax, we can drop this subject from the data analysis. Only 9 subjects was included in the final data analysis. According to bioequivalence statistical guidance, a minimum of 12 subjects should be included for the BE determination. Moreover, for the remaining 9 subjects, there is an imbalance in the distribution of the subject in the 2 sequences. 2 subjects from TR sequence and 7 subjects from RT sequence. It indicates a potential imbalance of carryover effect between the test and the reference products. Case 5, Tlag difference. The Tlag is defined as the time taken for a drug to appear in the systemic circulation. The indication for drug C is for adjunctive therapy for primary generalized tonic-clonic seizure and partially onset seizure with or without secondary generalization. The Tmax for this drug is about 4 to 11 hours. The half-life is 24 hours. It should be noted, for this study, Tlag values are consistently longer for the test product than for the reference product. Here is the PK profile for one of the subjects. You can see the Tlag for the test product is 24 hours. The Tlag for the reference product is 2 hours. So this is the distribution of the Tlag difference between test and the reference are summarized in this table. It indicates more than 50% of subjects have Tlag difference more than 6 hours, and 26.8% of the subjects had Tlag difference more than 10 hours. This raises a concern at the translation from reference product to test product at the study state. Due to the prolonged Tlag for the test product, you will see a decline of the drug C concentration when you switched from reference product to the test product. So this concern was tested using simulation approach. This raises a concern that in the subgroup of the patient, they may not therapeutic equivalence due to the drop of the drug concentration C when at the translation from reference product to the test product. It may lead a threat of seizures breakthrough due to this drop of the drug C concentration. So after consult with the clinical division for prolonged Tlag issue, the patient may experience subtherapeutic drug level when dosed for the naive patient with the test product or when switched from the reference product to the proposed test product. So they conclude the test product may not be therapeutically equivalent to the reference product. The applicant was asked to consider reformulating their product. Case 6, for lictin transdermal patch. Per product-specific guidance on this drug product, adhesive tape should be avoided throughout the study. When you see this graph, more than 75% of the patch age and a significant portion of the patch were covered with the tape. Since the tape could produce certain changes for the physical properties, such as the local temperature of the skin, moisture trapped under the patch, and the lictin evaporation rate. The applicant was asked to provide evidence to justify the using of the taping for the entire of the study does not impact for the BE determination of the test product under normal clinical condition use. Here's a case 7. This is an example for the unacceptable reference scale BE approach. The study was designed as 4-way crossover. You can see the within subject variability for the reference product for all PK parameters are greater than 0.294. Therefore, the reference scaled BE approach is used for the data analysis. You can see the 95% of upper bound are all less than 0. However, the point estimate for all the PK parameters are not within the 0.8 to 1.25. So this study was found inadequate. To summarize, to avoid the common BE deficiencies, it's important that we consider the design the study according to the PK property of the drug product. The agency recommends applicants refer to the product-specific guidance and the PK endpoint guidance. If the applicants propose a different approach from the product-specific guidance, please include adequate and scientifically sound justifications in the summary. Okay, here's the last slide from acknowledgement for their Truman, Larufa, April, Ethan, and Visual to help to tie up the slides. Thank you.